polymers form spontaneously without the help of enzymes or ribosomes. But unlike proteins, the polymers are a complexion of linked and cross-linked amino acids. So basically, it's possible that such polymers may have acted as a weak catalyst for a variety of reactions that took place on Earth. And then we move on to protobionts. Okay, so two key properties of life are accurate replications and metabolism, and neither can live without the other. So DNA molecules use and carry genetic information, including the instructions needed for self-replication. And the replication requires elaborate enzymatic machinery as well as nucleotide building blocks. And even though Miller-Urey's experiment has produced nitrogenous bases of DNA and RNA, they have never been able to create anything like a nucleotide. So,
Interesting.
self-replicating and metabolism appear together, and the necessary conditions for this that take place could have been met by protobionts. And a protobiont is a collection of abiotically produced molecules surrounded by a membrane-like structure. And many probionts show the proper ties in life, including simple reproduction and metabolism, as well as maintenance of internal chemical open environment from their surroundings. Laboratory experiments show that probionts could have formed spontaneously from abiotically produced organic compounds. An example is there are certain membrane-bound droplets called liposomes, and it can form from lipids or when other organic molecules are added to water. So, the first genetic material was most likely RNA and not DNA, and it has been found that RNA, which plays a central role in protein synthesis, can also carry out a number of enzyme-like catalytic functions, and their catalysts are known as ribosomes. And natural selection on the molecular level has produced ribosomes capable of self-replicating in the laboratory. And this is able to happen because RNA, unlike the double-stranded helix of DNA, they assume a variety of specific three-dimensional shapes mandated by their nucleotide sequence. And
Now, has there been any studies to show how that first RNA molecule actually came into existence?
not that I know of, or not that I read.
Okay, I haven't found anything either, so I didn't know if maybe you read something I didn't read or something, I don't know.
No, not that I read.
Yeah, that's the one hole in this whole thing is, we can, in a laboratory, we can make organic molecules without enzymes, abiotically, without living things.
Mhm.
But the problem is, I have not found any evidence that we can make abiotically an RNA molecule, because that carries a genetic code.
Well, what the book said was that when you compare RNA and DNA, because RNA is single-stranded and DNA is double-stranded,
Right.
it said that they assume that RNA, just like natural selection, it evolved over time and changed to a stronger double helix that we have
Right,
with DNA.
that I agree with. That I can understand completely. RNA likely was the first genetic material prior to DNA because it's the working molecule. DNA is just reference.
Yeah.
But making that first RNA molecule, good luck with that one. Yeah, I haven't found any evidence to suggest how that could have happened, but
So
wasn't sure if you had found anything.
everything up until making nucleotides is like possible.
Yes, but it's those nucleotides. Good luck trying to make one of those in the laboratory without living things.
Okay, why is it doing that? Alright, so in a particular environment, RNA molecules with certain base sequences are more stable and replicate faster with fewer errors than other sequences. And the RNA molecule whose sequence is the best suited for the surrounding environment and has the greatest ability to replicate itself will have the most descendant molecules. And the descendant molecules will not be a new species, but instead a family of sequences because of a copying error. And it is believed that this similar selection may have taken place on early Earth.
So with the fossil record, the significance of the fossil record is that it allows us to kind of trace evolution of life over time and to reveal major changes that might have happened in like the biology of an organism. And so the best source for fossils is sedimentary rock, and which is the rich. And so how we kind of do this, sedimentary rock, it's layered. It's rock layers that are called strata, and so a lot of fossils are found here in these types of rocks, and also you can also, like, fossils can also be found, like, insects can be preserved in amber, or mammals can also be preserved in ice. And so they show that, like, the really big changes that happened, like the big evolution that's happened from when we were just, when we had single cell organisms to like all the way through now we can see fossils, and how organisms and mammals and how they all progressed. But even though the fossil record is really significant and it has a lot of samples to look at, it's incomplete, and we do not have like a full, you can't see the complete pattern of evolution because there are definitely holes in the system. And so, and because of this, like most animals, like they haven't died at the right time in the right place to be preserved. So, a lot of the times the fossil record is really biased towards animals that were really prominent and that there were lots of in lots of different environments, a lot of times with hard shells as well because those are easier to be preserved. And so there's still all the time gaps in the fossil record continue to be filled by new discoveries, and an example of this would be how we found whale ancestors with hind limbs, and so that lets us know that previous to now, where now they live in the ocean, previous to now that they walked on Earth, and that through some process, through evolution, that they were more suited to living in the ocean than they were living on Earth. And so, how these fossils and these rocks are actually dated, the strata, the place in, the place in the rock in which the fossil is stored, it does tell us something about at what time the fossil was from, which is like your relative age. It gives us an idea, but it doesn't give us like an absolute date, like a really narrow time frame. And so one way that they try to date fossils would be radiometric dating. And so what this is they base this off the decay of radioactive isotopes. And so a parent isotope is just the isotope on its own, and then this parent isotope is going to decay to become a daughter isotope at a constant rate. And so the rate of decay, the way that they express it, is by half-life. So the half-life of an isotope would be how the time that it requires for 50% of the parent isotope to decay. And so each isotope has a characteristic half-life that's not affected by temperature, pressure, or environmental variables. And so an example of this would be carbon-14. It decays relatively fast with a half-life of 5,730 years, whereas uranium-238, it decays extremely slowly with a half-life of 4.5 billion years. And so the way that they do this is that isotopes of certain elements, they accumulate in the organism while the organism is still alive. And so an example of this would be carbon in an organism. It develops while this organism is still alive. So for example, like the most common carbon would be carbon-12, and the radioactive carbon isotope would be carbon-14. And so when this organism dies, it's not going to keep accumulating carbon, and the amount of carbon-12 over time is not going to change. But the carbon-14 that the organism contains at death, it's going to slowly decay into nitrogen-14. And so by measuring the amount of carbon-14 to nitrogen-14, the age of the fossil can be determined. And this method can work with fossils up to 75,000 years old. If they're really any older than 75,000 years old, most fossils are not going to contain the right isotopes that have such a long shelf life like uranium-238 to be able to be dated this way. But for 75,000 years, I mean, it still accounts for a significant amount of the fossil record. John?
Why is that?
What do you mean, well, why?
It's only 75,000 years.
Okay, alright, so looking back, carbon-14, right, that's something that a lot of organisms have. Its shelf life is, 5,700 30 years. Okay, so slowly, you know, after the 5,730 years, I mean, it's still decaying, but after a while, there's just not enough left. There's not enough left for scientists to be able to compare numbers with such a tiny amount. Like when the example is like most organisms, they're not going to contain uranium-238, right? But it has a shelf life of 4.5 billion years. So, you know, if you check it 75,000 years from now, if you check it several million years from now, there's still going to be a significant difference for them to be able to date it. Maggie?
What makes the carbon-14 turn into nitrogen-14? Like, does it just decay and it just does naturally, or?
Yeah, I think it has to do something with, the book didn't go into it, but like, I think it just has something to do with over time how the atoms are like getting eroded.
Yeah, when radioactive elements give off energy, they also give off particles, which include both protons and neutrons. If you change that proton number, you change the element.
Yeah.
Okay, that's the key. So, you know, carbon has,
6.
6 protons. Nitrogen has what?
5? Is it 5?
Is it 5?
I think it's 5.
I thought it was
I thought
7.
it was like about
So then how does it
It's
decay?
7.
So then how does it?
That, I don't know. That's something that, if you could look into that and see what it is, how did it change? Because I know if you change the proton number, you
You have
change
to
the
change
element.
the element, right?
Right.
So how is it turning from carbon to nitrogen?
Well, the only thing is, like, you would think it has something to do with pressure, but a lot of these isotopes really aren't affected by
Right.
pressure or
Yeah, we should
temperature
look
or
into that.
environmental variables.
Wait, what makes something radioactive? Is it just
If it has extra neutrons beyond the proton number, it's a radioactive isotope.
What does it mean to be radioactive though?
Radioactive means that it's gonna give off energy in the form of electromagnetic radiation. And it's on the higher energy end of the spectrum beyond the light range, beyond the visible light range.
Doc, I thought that isotopes didn't have to be radioactive.
Yeah.
Isotopes are just variations in the neutron number
Right.
of the same element. So when you have X number of protons, you have a specific element. But you can have examples of that element having variable numbers of neutrons,
Right.
which makes them
Give up energy.
of a different weight, and therefore they are isotopes. Generally, if you have more neutrons than the proton number, then it's radioactive.
Radioactive.
With the exception, of course, of hydrogen, which is only a proton and electron.
Okay.
But I would like you, if you could, to look into that. And let's examine how does it become that.
Okay.
We should look at that.
Okay. So, keep going. So determining the age of fossils in sedimentary rock can be difficult. Like I said, one reason is because most organisms don't have isotopes with long half-lives, that with isotopes that are like in bones or shells, so they don't have the ability to be stored after this, like the 75,000 years. But also sedimentary rock, it's composed of different sediments of different ages. So like, just because you find a rock in one layer, it doesn't mean that layer is only pertaining to one age. It can pertain to a different age, and so therefore you can't just usually just date them directly. And so a lot of times what they'll do is if like a fossil is found between two different layers, what they'll do